So we're going to do a real quick one here, just looking at major histocompatibility, complex proteins, and recognition of self. MHC proteins, there's a number of different classes of these, but mainly we're going to look at 1 and 2 classes, also sometimes known as HLA proteins. What these do is they play a role in the immune system either recognizing self versus foreign, and they also play a role in activating some of the T cells and B cells that are present in the body. What's kind of interesting is all the lymphocytes we're going to talk about, it's about 500 genes that are used to kind of do this whole immune system, and it's really a combination of using certain genes within certain classes to get these combinations of receptor subtypes that are going on there. And a lot of this recognition on the T or B cell receptors is by these MHC proteins providing that antigen to those T and B cell receptors. The other thing that's really neat on this one is it's all pre-programmed. We have a number of different combinations that are possible. Pretty much what the body does, and we'll talk about this on one of the next shows here, pretty much what the body does is it tests all these different combinations, and any combination that responds to a self-antigen, that combination genetically is pretty much basically deleted, so we don't make that. So what you're left with is any antigen specificity that is not self that the immune system can respond to. And each different lymphocyte, or all these different lymphocytes, have different specificities. And really when you have a particular lymphocyte that recognizes its specificity, that's when it's going to bring about a response. And again, do a few things with that. You're going to get some clonal expansion, setting aside of memory cells, things like that, that are going to have this happen. But the main thing we have to see here first is most of these cells, B cells can actually bind directly to the antigen, but all the T cells actually have to have some type of presentation of this antigen. And that is what these MHC proteins are doing. They are basically displaying the antigen for these T cells to bind to them and see if they're specific or not. And again, for these T cells, what you need is some type of antigen presenting cell. And in reality, any cell that has a nucleus in the body does act somewhat as an antigen presenting cell, but we're going to see that there is some specific antigen presenting cells that they are what I would call the professionals. It's their job to do that. Things like dendritic cells, macrophages, and B lymphocytes, that is part of their actual job, where a lot of these other ones are just announcing what antigens they have in them. And again, how this works is it's by these MHC proteins. These are specialized proteins that can take proteins and present them on the surface of the cell for these immune cells to take a look at and see if they respond to. So we have what are called MHC1 that are found on any nucleated cell in the body. And then we have MHC2 that are specific to those antigen presenting cells, or APCs, in addition to the fact that they also have the MHC1 class. So those ones that have both 1 and 2 on, those professional APCs. What's going on with MHC1 proteins is these ones, I mean, for each person it's genetically distinct to you, but what these do is, as we make proteins, these MHC molecules are in the membrane, and what's going to happen is you make these proteins, random parts of the proteins that are your self proteins are going to bind to these particular MHC receptors and then get brought to the plasma membrane. So really, when you look at these MHC1s, they're taking a piece of the cell that's being made, or a protein with the cell that's being made, and said, here's what I'm made of. So these are really presenting self-antigens on the surface of all these cells. Every once in a while, if a cell is infected, it would end up presenting, like let's say, a virus protein, and that's how sometimes the immune system can respond to cells that are actually infected as well. But normally if they're displaying self-antigens, they would be ignored by the immune system if your immune system is functioning correctly. And this little thing here shows the idea. You're making these fragments. As they get made, bound to these MHC proteins, they're going to get targeted to the cell membrane and displayed by going through the Golgi. Like I said, during an infection, what's going to happen if you get a cell that's infected with a virus and you're manufacturing viral proteins, this is also going to present those. So when a cell gets infected, these MHC1s would present that, and it could actually trigger an immune response by these infected cells by, let's say, a helper T cell or a cytotoxic T cell, which we will talk about in the next show here. And this is again showing that idea with an unhealthy cell. There's something in here breaking down the viral proteins. It gets into a piece of this here as you're manufacturing it, and that viral protein gets put on the surface instead of a foreign antigen. The immune system responds to that, sees it, and then is going to try to actually attack this cell. The other thing we have going on with MHC proteins is MHC Class 2. Like I said, these are on those professional antigen-presenting cells. These are things like I said, dendritic cells, macrophages, for example. These ones, when they engulf something, so macrophage, when it eats something up, or dendritic cell, when it takes something in, it's going to have whatever it engulfs fused with that lysosome, that digestive enzyme-containing organelle within the cell, and it's going to break apart pieces of this foreign protein. It then does what I always joke around as seafood, where it's like it chews it up and it's like, nah, seafood, and it puts it on the surface, so it's shown what it ate. This is another way that foreign antigens are exposed to the immune system. So this is showing the immune system that these phagocytes found something foreign, broke it down, digested it, and here's a part of it, and that also can trigger this specific immune response. And again, it's a way for helper T cells to get activated. And again, showing you the idea here, you ingest phagocytize some type of bacteria. It gets taken in, broken down by the lysosome. Pieces of that are then bound to that MHC Class 2 and displayed to the surface of the cell. And again, it's once these things are bound, these antigen-presenting cells, along with some of these other receptor subtypes on these T cells, this particular cell presenting it, it can bind to it here. And if, again, it was a foreign antigen and it was specific to this, that is going to cause this T cell here to become activated and bring about an immune response, which we will talk about the next time. And really how this all works, and we'll see on the next show here, we have a lot of different T cells with a lot of different specificities. What we end up doing, as I was saying before, is anything that does respond to self, we tend to delete those particular clones, but then when we actually do have a antigen come in there, if it binds to a particular cell that's specific to it, we are going to get a clonal expansion, this multiplication of the cell in a setting side of memory cells, which can then bring about a secondary response the next time we see this antigen. So the next time, like I said, in the next show here, we'll talk about some of the different, how the T cells and B cells are activated and what they do because of that.